great pleasure and an honor to introduce for you Professor Pierre Corvol, who will receive the Leonardo da Vinci Medal of this academy, which is the highest honor, as you know. But Pierre Corvol is not a chemist. He's a medical doctor and a scientist. After an internship in the Paris hospital and a postdoc with the Bethesda National Institute of Health in the US, Pierre Corvol became associate professor at the University Hospital Center Brousset Hôtel Dieu in Paris and later director of the INSERM unit on vascular pathology and renal endocrinology. INSERM is the French National Institute of Health and Medical Research. He was elected in 1989 to the chair of experimental medicine at the Collège de France, the most prestigious academic institution in France, which was founded in Paris in 1530. This prestigious chair was once occupied by Claude Bernard, the founder of experimental medicine. Pierre Corvol was president of the Collège de France from 2006 to 2012. He is now Professor Emeritus and Honorary Administrator of this institution. As both a scientist and a clinician, he has made pioneering contribution to the understanding of the role of the renin angiotensin system in human cardiovascular diseases and their treatment by blockers. This hormonal system plays a major role in the control of blood pressure, cardiovascular resistance, and regulation of water and salt metabolism. It is involved in many pathological diseases, such as high blood pressure, cardiac insufficiency, the presence of excess proteins in the urine, and the renal dysfunction in diabetes. The blockade of the renin angiotensin system by inhibitors has revolutionized the treatment of these diseases and has saved millions of lives. Pierre Corvol's research is paradigmatic of what is called today translational research. Beyond his brilliant scientific career in the medical sector, with discoveries of considerable relevance to human health, he has been instrumental in advocating for ethics and research integrity in science. For example, he has triggered their implementation in the training offered to students and researchers in doctoral schools. Ethics and research integrity are also of broad international relevance to open science and publication policies. Pierre Corvol's report on the French government, on scientific integrity has attracted considerable attention and led to many public initiatives. Pierre Corvol has been scientific advisor to various prestigious institutions. No way I can list all the functions and responsibilities as held. It would take me at least half an hour. His numerous awards include the Alexander von Humboldt Award, the Tigerstedt Award from the American Society of Hypertension, the MS International Award of the International Society of Hypertension, the Grand Prix of the Research from INSERM, the Grand Prix de la Fondation for the Medical Research in France, the Medal of the Frey-Verley Foundation, just to name a few. Since January 2019, he's president of the French Academy of Sciences. Dear Pierre, in recognition of your outstanding contribution to the development of major branches of medicine and your instrumental role in advocating for ethics and research integrity in science, the European Academy of Sciences is delighted and honored to present you with its highest award, the 2019 Leonardo da Vinci Award.
To receive this prize, and really, I consider it as a recognition not only of my work, but really of the team I've been directing both in hospital, clinic, and also in the lab for almost 50 years now. So it's really a lifelong achievement which I'm going to report to you today. And I'm very proud because after all, this is the 500th anniversary of Leonardo da Vinci. So when I told to my friends I was receiving this award, they were extremely, extremely proud for me. So I'm proud for me, yes, especially because Leonardo da Vinci, as you can see on this slide, worked on the heart, especially on the functioning, not only on the anatomy of the heart valves, anatomy of coronary vessels, but also of the dynamics of blood vortices which were in use. So it's quite interesting to see how, you know, at this time, he was already able to combine both anatomy, but also blood dynamics. And in fact, it's a bit what I'm going to report today with a lifelong journey into the renin-angiotensin system. Everything started a long time ago, and it was relatively recently that the renin-angiotensin system was rediscovered. Because as you can see, in 1970, the renin system was only supposed to be involved in renovascular hypertension. Then after, with fundamental research and with pharmacologically good inhibitors, the role of this system became more and more clear. And nowadays, I think people of our age, there's a good chance that some of you might be taking one of those drugs I'm going to describe. So in bold, I've just written what our group has been contributed to, cloning of the renin-angiotensin system components in order to understand the function of every element of the system. And of course, we have started genotype, phenotype studies in humans on the renin-angiotensin system genes. We have worked on renin inhibitors. We have produced genetically modified mice in order to understand better in depth the function of every gene. And we have started the first work on genetics of human hypertension. You have a oversimplified scheme of the renin-angiotensin aldosterone system. This system is crucial for maintaining blood pressure and blood volume. And you have here a feedback mechanism in which you can see that blood pressure, for example, down-regulates the renin-angiotensin system when blood is expanded or blood pressure increases. And the renin system acts directly on cardiac and renal function, but also through the production of aldosterone, which is a mineralocorticoid, it's a steroid hormone, which acts on the kidney. There is also a short feedback loop with angiotensin 2, which is produced by renin acting on its substrate, which inhibits the production of renin. Now, the cascade of events has been elucidated something like 50 years, 55 years ago already. Renin is an aspartyl protease which cleaves angiotensinogen, which is produced by the liver. It's quite interesting because renin acts on a single substrate, which is angiotensinogen. And it produces the N-terminal peptide of angiotensinogen, which is a decapeptide, angiotensin 1, which is inactive by itself. It has to be converted into an active peptide, angiotensin 2, octapeptide, through the action of the angiotensin-converting enzyme, ACE, which is a carboxyl dipeptidase. Now, angiotensin 2 and angiotensin 3, which differ just by the aspartyl residue, which is lacking at the N-terminus of angiotensin 3, and these two peptides are equally active and act on 81 angiotensin 1 receptors, which are seven transmembrane receptors, G-coupled receptors. The main effects of this system are, as you can see here, vasoconstriction, aldo production, thirst, sodium retention, activation of the sympathetic nervous system, which ends to, when it is overproduced or overactive, to high blood pressure and to organ damage, especially the heart and the kidney. So this system, as I said, can be blocked at every step by different drugs. The first one which were described by Miguel Ondetti and David Kochman in Bristol Myers Squibb in the late 70s. Captopril was supposed really to be one of the very first rationale drug design, quite interesting, and enalapril after by Merck. DuPont, de Nemours, and then Merck developed AT1 receptor antagonist, roughly 10 years later, and relatively recently, renin inhibitors, aliskiren, was developed by Novartis. When I started really to be interested in this field, I didn't know, and nobody knew, that in fact blocking the cascade of the renin-angiotensin system could be so beneficial for patients, because you can decrease blood pressure, you can reduce left ventricular mass and proteinuria. There is a reduction of cardiovascular morbidity and mortality, which has been proven by numerous studies. There is a decrease in the progression of renal insufficiency. We do not have any drug, actually, to stop the renal insufficiency, especially in diabetics, besides those drugs. And a reduction of cardiovascular events in patients with a high cardiovascular risk at baseline, which means that even in a patient who has no increased blood pressure, if there is any cardiovascular risk, one can discuss of the prescription of renin-angiotensin blockers. Now, when I started, as I said, very few things were known about the genes. So the first idea we had was, of course, to try to elucidate the structure of renin. In the 70s, the only way to do it was really to purify renin. To purify renin is easy to say, but it's extremely difficult to do because it's only 1/500,000 proteins in the kidney. And therefore, we started with affinity chromatography, which was just being developed by Quetric Assas in NIH, where I was and discussed with him. So the first renin inhibitors, which were very weak, were in fact synthesized by Pedro Quetric Assas. We were trying to find another type of inhibitors, and we were interested by the discovery of Umezawa and Aoyagi in Tokyo of peptide, which is a universal aspartyl protease inhibitor. You can see here in red statin, this statin, which is an unusual amino acid, which acts as a transition state analog of the catalytic site of renin, the second one which is indicated here. So peptide could be coupled to cephalose to purify renin. We started with renin. We had 70 kilograms of renin to produce minute quantities of renin, which, after all, we could not do very much with it because there was no microsequencing available at this time. But sometimes you have to have luck. And luck came from the clinic. One of our patients had a very severe high blood pressure, which was due to a very, very, very rare disease due to a renin tumor. You can see the renin tumor here. In the CT scan, you have this hypodense tumor. So after making the diagnosis, which was not so easy at that time, we extracted the tumor, and from the tumor, we made cell culture, we made antibodies, and we made cloning. That was really something which helped us a lot. And we, by cloning, we discovered that renin was indeed an aspartyl protease. You can see here the signature of an aspartyl protease with the tetrapeptide indicated here in this, the two tetrapeptides face together in X-ray structure to make the active site. And what was interesting also was the fact that renin is synthesized as a precursor. Remember, it's an aspartyl protease, so like the other aspartyl proteases, it is synthesized as a zymogen. So pro-renin, this pro-fragment, inhibits the action of renin. Actually, you have made some peptides from the pro segment to inhibit renin in now early inhibitors, renin inhibitor search, and you have a pre-peptide which is cleaved off. Now, when we cultured the cells from these renin tumors, we observed that renin was produced in granules, in mature granules you can see here. In fact, it is packaged here, and renin is processed in these granules to produce the active renin. But you have also the proto-granules, which will become then after the mature granules. You can see also myofilaments here, which shows that the renin justaglomerular cell is a peculiar cell because it's both an endocrine cell, this is typical of an endocrine cell, but it's also a cardiovascular, it's a vascular smooth muscle cell, as shown with these myofilaments. And if you look now about double labeling of by double labeling at the electron microscopic level renin, you can see these big dots, which are renin itself, and the small dots, which are here, but better seen here, which are pro-renin labeled with the pro-renin antibodies. And we proposed the following scheme for the production and secretion of renin. You can see here that in justaglomerular cell, renin is synthesized as prorenin. Prorenin can be exported as it is, inactive renin, in the blood, but it can also be packaged in these granules, mature into renin, and also produced in the blood. So in the blood, you have both an inactive form of renin and an active form of renin, which made us to suppose that renin is both an enzyme, it's clear, it cleaves angiotensinogen, but it's also behaving as a mask hormone, because this is typical of a pro-hormone production, which makes the story quite interesting. And in fact, this scheme, which we proposed a long time ago, is still quite the scheme which is accepted. And we could, with the antibodies, for example, show the presence, you can see here, the immunolabeling of renin, which is just beside the glomeruli in this special part of the afferent arteriole leading to going to the glomerulus, and you have here baroreceptor sensing for modulation of renin production, as well as innervation by the sympathetic nerve system. Now, the second enzyme in which we were quite interested was a converting enzyme. Converting enzyme, as I said, is a dipeptidyl carboxy peptidase. We cloned the human and some other animal species converting enzymes. And as you can see here, it is transcribed from a gene in which you have two active sites which act independently, which are not cooperative, and it results from a gene duplication. This is a somatic enzyme, 2 active sites. Of course, it was quite interesting to see if those 2 active sites were strictly similar or not, if they had any sort of specific substrate, and if we could design specific inhibitors of either site, what we call the N-terminal site or the C-terminal site. There is also a testicular enzyme, which is produced, again, from the same gene, but with a specific promoter, a germinal promoter, which codes for a single active site enzyme. And the converting enzyme is inserted into the plasma membrane by a short transmembrane domain, which is indicated here. So we knew that, of course, this enzyme was able not only to cleave angiotensin 1 into angiotensin 2, but was also able to inactivate bradykinin. And therefore, when you inhibit the converting enzyme, you block the production of a vasoactive, constrictive pressure peptide, but also you potentiate a vasodilator and hypotensive peptide. Now we, with Mario Capecchi and with Ken Bernstein, we have made a series of genetically modified mice to identify the role of converting enzyme, blocking, for example, one side, the other side, or the 2 sides. When you do this type of experiment, sometimes you find what you expect. Blood pressure decrease, that was already shown actually by pharmacological treatment of animal and in the humans. And in fact, if you suppress the converting enzyme completely, delete the gene, you have a blood pressure reduction, you have a gene dosage effect because you can progressively suppress one or the 2 enzymes. You have also modification of kidney morphology. You have some renal insufficiency. What was not expected at all was we produce anemia and male infertility. I'm not going into details, but this raises obviously a quite interesting question. We are not yet completely solved at the present time. I'll skip this one. Now, I was always interested in evolution, and the renin-angiotensin system is a good model for evolution because you will see mark the appearance of the system with the vertebrate and the passage from water to earth is probably very much dependent on the functionality of the system. You can see here the presence of the renin-angiotensin system in all the vertebrates, starting with the early vertebrates, and you can see that the action of angiotensin 2, the octapeptide, is extremely conserved. There are some minor variations, but which are not important in terms of stimulating the angiotensin receptor. And you can see here that you have a vasopressor action all along the phylogeny of the system. You have an action on steroids production, an action on the sympathetic nervous system by catecholamine release, a central action. We search all these actions come together to control blood volume, regulation of water and salt, and blood pressure, cardiac function. And therefore, it's not surprising this system is very important in terms of the passage from the seawater to earth. And you can see the angiotensin converting enzyme is the enzyme which is the oldest of the family, but all the other parts of the renin-angiotensin, the other genes, appear roughly between 450 500 million years ago. With some of our colleagues, we have worked on the evolution of ACE. I'm not going into detail, but we were able to find ACE in protobacteria. Of course, there is no blood pressure regulation. There is nothing of this sort. But it's interesting to see that probably the very ancient function of ACE was to act as a peptidase, a final peptidase involved in final degradations of proteins. We did not find ACE in plants, but as I said, in this Xanthomonas C3 bacteria. And surprisingly, the active site is the same. It is inhibited by captopril, which was designed in mammals. So you can see the conservation, even though there are no other elements of the angiotensin system in bacteria, in annelids, or even in insects. In insects, in invertebrates, you find ACE, several ACE isoforms actually, but no renin, no angiotensinogen. Now, the big question is, and I will end up with that, can we live without a renin-angiotensin system? This is quite important because, after all, if you take a renin blocker, an angiotensin converting enzyme blocker, an AT1 blocker, can you push the dosage? Is it dangerous or not? Can a pregnant woman take ACE inhibitor or not? We can give the answer. Yes, if you are a mouse, yes, you can live. Not very well, but you can. You can because you do not have abnormal development. The fetus is apparently normal. However, at birth, you may have difficulty to thrive. You need salt supplementation. There are some blood pressure decrease in adults, not negligible, and a renal insufficiency, but life is possible. What about human? In humans, you cannot live, at least during fetal life. That was a discovery made by my colleagues in Necker, Olivier Gribouval and Marie-Claire Gubler. They had several families, there are now more than 120 families, in which we observe a defect, a loss of mutation, a loss of function of one of any of the renin-angiotensinogen genes. Either angiotensinogen, the ACE, renin itself, or the AT1 receptor. If they are mutated, if they are inactive, there is a very severe disease during fetal life, which is called renal tubular dysgenesis, which is a disease which happens at the end of the pregnancy. It is an early anuria, a stop. Renal function is stopped, and they are the same phenotype regardless the gene mutated. So again, whatever is a gene mutated, you have the same phenotype, which is interesting because it shows that there is no redundancy in the system, which means this system is absolutely crucial, at least for early development. Now, we were quite interested because, of course, looking at the mutation, it could give us some ideas how the renin worked, how ACE worked, etc. It was really a sort of replication of what we like to do in vitro site-directed mutagenesis. Here, it's an in vivo mutation which is responsible for the inactivation of the enzyme. I'll just take two examples, one on renin and one on ACE. On renin, there is a single mutation, where there is a replacement of a tyrosine residue by a serine residue. At the beginning of this hairpin, you can see in yellow, which is called flap. The flap is a mobile hairpin structure which fixes the substrate, the renin substrate, within the molecule. And this disruption due to this mutation is able, as you can see here, in 2 families to induce perinatal death. There was just 1 living patient because we knew that his brother died of this mutation. So he went on peritoneal dialysis and transplantation at a very early age. And we could see that by this in vitro culture system I showed you before, that prorenin was still synthesized in cell culture, but was not able to be exported. So there was a modification in the folding of the protein, which, inhibited its exportation. That's the first case. Now, the second case is even more interesting for us because, as I said, you have here a scheme of the tissue-bound angiotensin converting enzyme. You have a short intracellular part. The C terminal is indicated here. You have the N domain of ACE, the C domain of ACE. The 2 domains are linked by what we call a bridge region. We were wondering, and as I said, there is not only a tissue-bound ACE in endothelial cells and epithelial cells, but there is also a circulating ACE, which you can measure. So there is a shadase, which we don't know yet exactly what it is. It's probably an ADAM-like enzyme, but this shadase is able to produce a soluble ACE you can see here. The question we asked ourselves was, is the circulating ACE important, or the tissue-bound ACE important, or both? So to solve this question, what we did was to make a side-directed mutagenesis by introducing here a mutation which block the translation, and therefore we could not have any tissue-bound ACE, but only circulating ACE. And so, again, with Mario Capecchi and with Ken Bernstein, we produced this mouse in 1997, and we showed that this mouse had a low blood pressure and had an impaired renal function. It behaved exactly like a mouse devoid of any ACE, even though there was a circulating ACE. And we found, in this series of patients with these mutations responsible for renal tubular degenesis, we had almost the same mutations as the one we created 15 years ago. This mutation, which we could study, was reproducing exactly what we observed a long time ago with these genetically modified mice, showing that, in fact, it's quite interesting to do exactly as Pierre said, translational research. Actually, we anticipate, with some circle we are correct at this time, we anticipate what we found later on in humans. Now, I would like to leave you with a few conclusions. The anti-renin angiotensin system emerged about 500 million years ago. I believe it has played a decisive role for the water-to-earth migration, salt handling, and heat adaptation. Gene inactivation is lethal in human during fetal life. It seems not to play a major role during development. I cannot really develop on that, but the abnormalities we observe are rather due to a very low renal perfusion pressure in the fetus than a direct morphogenic effect of the system. I cannot really go also into detail, but we believe that there are some variants of the renin-angiotensin system gene, especially the angiotensinogen variants, which may have conferred a selective advantage at the origin of mankind. The blockade is beneficial, I showed you, but even in adults, if we force the blockade of the renin-angiotensin system, especially in water and salt deprivation, patients treated with high doses of diuretics during overheat, this could be harmful and reproduce to some extent what I've shown you before. But you know, 50 years is not enough to work on the renin-angiotensin system. I need 15 more other years because, because there are some other peptides derived from angiotensin 1 which might have some biological properties. There are other enzymes involved in angiotensin metabolism, other angiotensin receptors. I discussed over 81, there are not an 82 receptor, probably not very important, but still. We have been working on at the CNS level, where angiotensin 3 seems to be a key player. Actually, we have a drug in development in phase 3 now, blocking the conversion of A2 into A3, which is promising, I hope. It's a part of other renin-angiotensin system inhibitors. Of course, we dreamed at least, but we are not able yet to personalize treatment, because we have several now variants of the renin-angiotensin system in humans. So we're hoping to individualize those variants, those gene variants, with a response to renin-angiotensin blockers. This is not feasible at the present time, maybe later on. So this is in 75. This is my small group. I was interested because, you know, already we were advocating for a peaceful world, and we were concerned about pollution. This is it's not a Photoshop treated slide, right? And now the group, of course, has expanded. There are many more people who should thank at the end of such a talk. I thank you very much.
Well, thank you very much indeed, Pierre, for this brilliant lecture and for convincing us that this kind of research needs time. But the achievements are already very impressive, and we're very grateful to you for the work done and for being with us this morning on the occasion of this Leonardo da Vinci Awards. Thank you very much indeed. If there is any question, we can handle one or two. Otherwise, we can have further time during the lunch break.
So now it's time to say farewell to all of you, and we hope to have all of you next year in Paris. So I think that now you know that we are getting to the close. We have the lunch. The ones that wants to go to the excursion can go to the excursions that have been already planned. I hope that we can account with all of you for improving better what we are doing, because we just heard that our distinguished and outstanding Leonardo da Vinci Award, he just said that he plans to have more 50 years. It means that we have to have a better planet if he wants to reach this goal. Anyhow, it was for us a great, great pleasure to have all of you. And just farewell. I don't know if you want any other remarks. And we keep in touch. And please make your contributions on Science Kind for our academy. Thank you very much to all of you.